Thanks for having me. This is joint work with Harris Wang, who is at Columbia Medical School, and with his former lab mate when he was in grad school, Michael Mead, who is at Harvard Medical School in Boston University. So, I'll start off by saying this is a pretty strange talk. This is a talk about behavior and a little bit about evolution, but it doesn't involve people, and not even primate people, other kinds of primate people. So this is a talk about the behavior of bacteria by an economist of all things. And actually, if I love interdisciplinary things. If I could go back in time 5 years, maybe 5 years into the past, and tell myself I was going to give a talk about the economics of microbial ecosystems to a bunch of anthropologists who study evolutionary psychology, I would be beside myself. I would be absolutely thrilled. But at the same time, I realized that this might be a little bit of a stretch for the interests of the audience here. So I hope to motivate this. I hope this is of interest to the crowd. And I think it might be of interest because even though the topic is going to be something that's maybe not so central to what people care about, the way we're going to think about this problem is going to use foundational social science modeling to understand microbial sociality. So conceptually, I think the frameworks that we're going to use are frameworks that might be useful to thinking about for evolutionary psychology, anthropology, etc. Okay, so I want to begin by just motivating the importance of microbial communities. So microbial communities are everywhere. So Craig Ventnor, who is a very successful microbiologist, who was instrumental in the Human Genome Project, he took sort of a vacation/research trip. He got a beautiful sailboat, sailed around the world, and they were collecting samples of seawater around the world, sequencing the DNA of the bacteria in the seawater. And they found just this plethora of biological diversity, just thousands of different types of species and bacteria that had never been discovered before, and lots of variants across the oceans as well. So it seems like there's these microbial communities that are basically everywhere. They're high up in the atmosphere, they're deep in the ocean, they're even deep in the Earth's crust. There's extremophiles that can live at high temperatures. So for example, areas like deep sea vents where the temperature exceeds 100 degrees Celsius. There's bacteria that survive in highly acidic environments. And one speculation of Craig Ventnor's is that some of these species are actually so resistant to desiccation and so resistant to high radiation. Now, it might be the case that some species, terrestrial species, bacteria, have made it all the way to Mars. And here's how the story goes. Of course, this is highly speculative. But the solar wind blows off matter from planet Earth. And some of this matter ends up in the gravity well of Mars, so it eventually reaches Mars. And the estimate is about, we exchange about 100 kilograms of matter with Mars every year. And so if you think about, over the course of 4 billion years that we've had life on Earth, it might be the case that some spore has survived the travel. Anyway, that's just speculation. But the point is that these microbes live everywhere, and these microbes are also very important. These are absolutely essential for our own human functioning, and they're also essential for the functioning of ecosystems. So we have 10 times more bacterial cells than human cells in our body. Our skin's covered with bacteria. Our guts are filled with bacteria. And even though that sounds like 10 times more bacteria than humans, it's not like we're like walking bacteria. Only 1 to 3% of that body mass is actually bacteria. These bacterial cells are much smaller. But we are sort of these symbiotic walking beings, and these bacteria in our bodies, these microbial flora in our gut, are absolutely essential for a variety of functions. So a lot of this research has been done at UCLA. So what I'm about to read to you is essentially headlines that I pulled off of NPR over the last few months. So they've done a variety of studies. One showing that the bacteria that we have in our bodies affects metabolism, affects obesity. So they found that leaner people have more diverse gut bacteria. And they've actually done experimental studies where they've had mice that were obese. They fed the obese mice bacteria, gut bacteria, from lean mice, and the obese mice lost weight. This is controlling for the diets of both of the mice. Microbial flora also seems to play a role in mental function. So they had, this is sort of one of the most surprising things. I didn't really believe it when I first saw it. But what they found is they found mice that have autism symptoms, and they effectively cured these autism symptoms by feeding the mice good gut bacteria. And then we also know that the gut bacteria influences immune system response like allergies, multiple sclerosis, etc. Microbial communities are essential outside of the body as well. They play an essential role in soil ecosystems, fixing nitrogen when it's necessary, which is necessary for agriculture, and they play an important role in the carbon cycle. So these microbial communities interact in complicated ways that are not yet well understood. In particular, there's a number of different behaviors that sort of manifest in these microbial communities. They form biofilms. They interact in various interesting ways. But one of these ways and the topic of today's talk is that these microbes crossfeed. And what that is one species will export metabolites from their intracellular space into the environment, and then other species will eat these metabolites. So there's this cross-feeding behavior, and as soon as I hear this, I think, as an economist, I think, oh, this is an exchange of metabolites. This is basically economics, right? You have different species that are exchanging resources, and effectively you could think, well, economists have been thinking about exchange and trade for, well, maybe about 300 years now, 200, 300 years in that range, and so maybe we could take some of the ideas from economics to help understand this particular system. Currently, there's no general framework to understand this. So what do? So a microbial consortium is analogous to an economic market. Microbes grow by trading with each other, just as countries grow by trading with their neighbors. And so general equilibrium theory is perhaps the most central model in modern economic theory. The idea behind general equilibrium theory is that given a set of consumers with well-defined preferences, and I'll go into the details of this model in a few slides. Right now I'm just going to give you an overview. So this is the core. This is sort of the core model in equilibrium, perhaps the most core model in economics. Every grad student learns this in their first year of the PhD. They learn general equilibrium theory, and the idea is that you have a group of consumers. They have these well-defined preferences. You have firms that have well-defined production technologies, and there's an initial endowment of resources in this system. And then what general equilibrium theory does is it determines the ultimate production and distribution of goods. So that's the purpose of this model. It's to understand, given this sort of mechanical world that we set up, what do people produce, what do they get in the end, and what are the prices that allow that to happen. Now this same sort of thinking can be applied to microbial system. Microbes have well-defined resource demands, which are analogous to preferences, and I'll talk about that a little bit more in detail. They have well-defined metabolic pathways, which are analogous to production technologies, and they have an initial endowment of resources. So the systems are similar, but they're not identical. we're going to adapt general equilibrium theory to the special circumstances of biotic markets. Our biotic general equilibrium theory is going to be a framework to study trade in microbial communities. It provides an a priori theory of crossfeeding, and it leads to several novel predictions. So there's two main components of this paper. The first is presenting the theory, here this biotic general equilibrium theory. The second component of the paper is to test that using a laboratory experiment. We're going to test the model by building a simple system of synthetic E. coli that are programmed to trade necessary amino acids with each other. So my co-authors are biologists, and they have this technology where they've gone into these E. coli cells, and they have the ability to essentially tune these bacteria to trade more or less. So what's been found is that these E. coli, they naturally export resources into the environment. They naturally just sort of pump out these metabolites that seem necessary to growth. And the first thing you think is, why would any optimizing bacteria waste perfectly good resources and get rid of them? Right? And we don't really know. There is no real answer to that question. And so we're going to try and help understand why this is the case. And our story is that it's going to be because there's mutual gains from trade. So these E. coli are programmed to export more or less of a given resource. And this manipulation allows us to test the comparative statics of the model. So the model is going to make predictions about the behavior, the growth of these populations as a function of how much resources these bacteria are exporting. And we're going to test those predictions by manipulating the export rate of these bacteria. So by the way, I should have said this in the beginning. So economists are used to getting interrupted. The norms for economists giving talks is frequent interruption. So if you have any questions, feel free to ask at any point in time.
Is there any evidence that the behavior is conditional, that individuals donate the substances only when others are doing it and that they stop otherwise? Is there evidence for this?
That's a great question. This is exactly the next step, the next line of research that we're thinking about. And we were talking about this, just an hour ago. So as far as I'm aware, there's no evidence that bacteria have sort of like think of it like a reciprocal altruism. But this system, and this is sort of one of the last slides I have, this system could potentially be used to look at other forms of mutualism. So for example, we think that maybe the relationship between terrestrial plants and our biscular mycorrhizae fungi, this is one of the most important mutualisms in ecology. Basically, the terrestrial plants transmit carbohydrates and resources to these fungi. The fungi trade phosphorus. And there has been a study that's shown that, so these roots sort of live underground, obviously, and there's a diversity of fungi for each plant. And it turns out that the plant knows how, somehow, to divert carbohydrates to the fungi that are providing the most phosphorus. So there is sort of like a reciprocal altruism system built into that mutualism. The mutualism we're going to consider here will not have any sort of reciprocal altruism. We're going to think of this as sort of the dumbest setup where the bacteria, they just export a fixed fraction of a particular resource. And we're just going to analyze the dynamics of that. The next thing we would like to do, and that seems to be the case, at least as far as how when my co-authors sort of noticed that E. coli were exporting these metabolites, that seems to be what they were doing. They're just kind of exporting a fixed fraction. Now, the next step we would like to do is exactly what you're saying. Program in a positive feedback loop where the more stuff you get from the environment, the more that particular cell might export. So that, once you have that positive feedback loop, and that might sort of build in sort of a reciprocal altruism, and prevent, invasion of mutant free riders. Okay, so why? Who cares? So this is a question that economists love to ask. Economists like to say, a project isn't worth doing unless it's worth 1% of GDP. That's like a good rule of thumb. It's actually really good evidence. It's actually a really good.
Anthropologists do.
What?
Totally on the same page.
Yeah. Well, it's a really good advice that I think that I got when I was a grad student and that I would give to grad students as well. The point is that it need not actually be, it's not that you have to point to something like, oh, this is 1% of GDP right here. It could be something that's like a fundamental aspect of human nature that affects a lot of things that might maybe ultimately add up to 1% of GDP. The point is, it's something important. Don't waste your time on stuff that's not important. So I hope to convince you here that this is important. There's three sets of, I think this has implications in sort of 3 dimensions. The first is ecological. Which species will form naturally in communities for mutual gain? Right? And so what we're going to find here is that comparative advantage, and I'll explain what that concept is. Comparative advantage drives sort of the degree of mutualism, the degree of mutual gain between interacting species. And so the ecological implication is this model will help us to understand which species will co-locate with each other and which species will form effective mutualisms, effective consortia, and which ones won't. Okay, so that's the ecological question. The second question is the evolutionary question. How will species adapt into a trade niche? Right? And so it might be the case you have a community, you have a consortia of bacteria, and they maybe have a particular need. They're particularly inefficient at synthesizing a particular metabolite that would help the consortia grow. It might be in the advantage of a particular species to fill that trade niche, because then it's an incentive of the consortia to feed that species, to keep that species going there. Right? So this framework introduces the concept of trade niches. As far as I'm aware, I don't think anybody has mentioned that before. And the third implication is the engineering question. How can we engineer organisms in consortia to maximize productive objectives? So for example, we might want to efficiently produce biofuel or waste remediation or something like this, and introducing a particular bacterial species might be less efficient than using a consortia that works in concert. And so knowing how to design the optimal consortia, or possibly engineering the optimal consortia by sort of programming species to share more or less with each other, this provides a framework to think about that kind of question as well. Okay. Okay, so now what I'm going to do is I'm going to explain general equilibrium theory in math-free English. So typically what, if I were to explain this to, in an econ talk, we would just say, oh, and here are the equations, and then those, you do the standard thing to solve those equations. So I'm going, and usually 2 people in the audience understand what's happening. So, fortunately, I'm forced not to do that, and hopefully I can explain it in such a way that everybody's on board with this. So I'm going to tell you how general equilibrium theory works in sort of a story point of using storytelling. So we have, in general equilibrium theory, you have 2 groups of actors. And remember, this is the canonical model in economics. So when economists think of markets, this is the first thing that they go to usually. So there's 2 groups of actors. There's consumers and firms. Consumers have wants and needs. They're endowed with resources that they can sell, namely their labor and time, to purchase other goods. Consumers have what we call a utility function, and this is an abstraction that represents the consumer's preferences, namely their feelings on whether a bundle of goods is better than some other bundle of goods. Okay, so what is a bundle of goods? So for example, so this is how it works, God creates, the economy sort of appears, right? And consumers appear on the scene with, deep in their soul, they have a preference relationship. And this preference relationship is an ordering over bundles. We take this entirely as a primitive. We don't explain where it comes from. And the ordering of bundles, the way that works is that, so what is a bundle? A bundle is like, for example, I could say a house in Westwood, a small house in Westwood, a Toyota, Corolla, 300 bags of oranges or whatever. Okay, that's a bundle. And then maybe you have another bundle, which is a big house in Rancho Cucamonga, a Lexus, and 1,000 bundles of oranges or whatever, or 1,000 bags of oranges. And so the preference relation tells you which one is better, which one is preferred by that particular consumer. And that's all it is. You have all these different bundles, and the preference relation, the utility function, the same thing, more or less, defines the ordering of the bundles for that particular consumer. Okay. So now what does the consumer do? Well, the consumer sort of looks to the market, sees what all the prices are, including the prices of goods that they sell. So they're endowed, most likely with something like labor, or maybe they're endowed with some other things. And they choose the best bundle given their preferences that they can afford. Okay, so they go to the market, they sell some of their labor, they get some income, they use that income to purchase oranges and houses or whatever. And they choose the best bundle that given the amount of labor that they can sell. Okay. So they're optimizing. Firms, their problem is somewhat similar. The major difference between firms and consumers is that firms don't have preferences. So what a firm is there are these entities, there are these agents, they sort of appear into this economy, and they have this technology. So whereas, in this part, God creates the consumer with the firms, with the utility function. Here, the firm falls out of the sky and has a technology. Where does the technology come from? We don't. It's a black box. We don't look at what it does or where it comes from. We say that a firm has a technology which is basically an input-output relationship. It takes in input goods, it transforms the input goods, for example, labor, raw materials, transforms those into output goods, for example, manufactured goods and services, and the technology says, how that works. So, for example, 3 hours of somebody's time plus, some wood equals a desk or whatever. So then firms see the prices, and they purchase and sell inputs and outputs in such a way to maximize their profits. So firms are also maximizing. They're maximizing their profits, and they do so given the prices that they see in the market.
Sorry, can I ask
Yeah.
a question? So who sets the prices?
That's exactly what I'm getting. Excellent question. Like, I didn't even plant that question. That's a great question. So who sets the prices? Where do these prices come from? This is sort of the missing part. They're looking up the prices, and they're optimizing given those prices. I haven't said where the prices come from. Ultimately, the whole point of this model is meant to predict final prices, the production of the firms, and the consumption of all goods. The last piece of the story is an answer to your question. How are prices determined? Well, the equilibrium, we define this idea of the equilibrium price, it's where supply equals demand, okay? And so the idea is, it's the price so that any buyers don't want to buy any more given that price, and any sellers don't want to sell any more given that price. If a buyer wants to buy more, given the current set of prices, and no seller wants to sell to them, then that's out of equilibrium. So then we would imagine that the price will go up a little bit higher, so that a few more sellers want to sell and a few buyers want to buy less, and you get to the point where exactly equilibrates. And that we define as the equilibrium price, and that solves everything. That solves the model, solves the distribution of goods, etc., etc. Okay. So, So here's a key point that I want to point out that's worth observing. Here the price is perfectly enforced if a person works for 1 hour, she'll get exactly 1 hour's worth of wage guaranteed. So when they look up the price, we assume that there's this like perfectly well-functioning legal system. There's no risk of a lawsuit here, that the employer reneges on the labor contract or anything like that. If a person goes in and wants a candy bar, they can't just grab it from the store and run. That never happens. There's no risk it'll ever happen. So everything that happens here is voluntary. This is a system of voluntary exchange. Everybody knows exactly what they're getting into for every exchange. They know exactly what price they're getting. They know exactly what they're buying or selling. And it just sort of happens magically. There's no enforcement issues, right? Now, you might be wondering, how does this actually work institutionally speaking? Are we imagining this is like a bazaar where people go from stall to stall and there's some haggling going on, or is this like an auction house with an auctioneer calling out prices? So the way economists think about it, I really want to know what your reaction to this is. The way economists think about it is it's institution free. There is no institution. It doesn't matter what the institution is. This is the solution. It presumes that whatever institution is at work, it'll somehow arrive at this solution. So in that sense, it's a very powerful concept. It also it makes it very falsifiable too. I mean, you could test these things. You could see if institution matters. And the institution does matter. But in any case, this is a nice sort of approximation, and we think that well-functioning institutions will achieve this sort of outcome. Is there a question?
Does the metaphor depend on like the metaphor of currency, like the metabolites being uniform in exchange between the different?
Yeah, that's a great question. So now I'm going to point out what the differences are between this the standard economic framework and the biological framework. There will be no currency in the biological framework. Prices will play a much less important role in the biological system than they will here. And that's basically because we won't get market clearing. We won't get equilibrium prices, so to speak. We won't have supply and demand necessarily equating in the biological system. Okay. So let me compare and contrast, and then I'll go into the details of what the biotic channel equilibrium system does from the point of view of a cell. So there's many similarities between this view of human exchange and microbial exchange with some important differences. So similar, cells were designed by natural selection to optimize their growth. So the wants and needs of a cell are the resources necessary for mitosis, right? So, the wants and needs of humans, this is based on whatever, their culture or their, genetic endowment or whatever. Here, cells have different wants and needs. That's what resources they need to grow. And so this works, the analogy is perfect. This implies that cells have preferences over different bundles, just like consumers do. Some bundles are going to be better than other bundles for the cell because some bundles allow for greater growth than other bundles. Also similar, cells are both like a consumer in this case, and they're also like a firm in this case. So you could kind of think the analogy in human markets would be it's like each individual owns their own factory. You can kind of think about it like that. So cells have a technology, which is metabolism, that allows them to transform input metabolites to output metabolites. So again, the analogy works really well. And also another similarity, there are going to be different species that have different needs, they have different preferences, and they have different metabolisms, different technologies. And so general equilibrium theory is well suited for understanding heterogeneity, and so is the biotic general equilibrium. Now, a big difference is that here, we're not in a voluntary exchange economy anymore. It's not like two bacteria go up to each other and they say, well, they look up the prices and they say, oh, it looks like arginine is worth like $5. I'll sell you an arginine if you give me $5, and they do this kind of trade. There's no system like that. Instead, the way the system works is it works more like a gift exchange economy. It works more like a bunch of individuals giving to a common pool and then taking the resources out of the common pool. So what happens in our system is that cells are programmed by natural selection or by engineers to export a fraction of their production into the medium, and then they take resources from the medium via diffusion. So these little guys are floating around in this soup, and they're just kind of spitting out metabolites, and then they float around and they eat up the metabolites that they need. And so the common pool is, well, it's literally a pool. It's the stuff that they're floating in, right? And so you have these metabolites that are floating around, and you could think of it more like, like common grazing land or something like that, and people contributing to some common pool like this. Another big difference is that markets may not clear. So there might be leftover production floating in the medium. It might be the case that all these bacteria are exporting more resources of a particular metabolite than are consumed for that metabolite, or vice versa. You might have a surplus of a particular kind of resource in the medium that's being consumed faster than it's being generated back into the medium. So you don't have this sort of market clearing condition, and as a consequence, the idea of an equilibrium price is much less important in this system. Okay, so now what I'm going to try and do, I'm going to try and sort of finish explaining, yeah.
Having some trouble understanding, you're talking about optimization and then leftovers, right? These things sound like, sounds like a contradiction. How do you...
Right. Why would there be leftovers if people are optimizing? Well, here would be a really simple case. Suppose some engineers go in and program these guys so that they all export exactly the same resource, and then there. Let's see, would that work actually? I'm not sure if that would work. Here's another example that would probably make more sense. It's the flip side, where that they would be absorbing excess resources from the medium. Right? So if the medium begins with a surplus of, I don't know, say arginine or some particular metabolite, they might use up this metabolite. And so the original endowment of arginine into the medium is going to decrease. So that doesn't actually answer your question, but that at least that shows that the equilibrium is not necessarily clearing. It's not necessarily clearing. You could have a decrease. Off the top of my head, how can you get a surplus? I feel like I've done numerical examples where they have been, but I probably won't be able to remember until after the talk. I'll try and think about it. Mm-hmm.
Just wondering, I mean, if you take the equilibrium out of it, that's one of the major differences, then why is this a general equilibrium?
Oh.
Is it just a leftover that this is the name of it? But I mean, it seems like the equilibrium is a really critical thing, and that's the thing that doesn't actually have a parallel.
Yeah, so we're going to, there is a sort of an equilibrium concept here. So each cell is going to be optimizing individually, and that's what I'm going to talk about on this slide. Each cell individually optimizes, given the resources it's getting from the environment, given sort of the constraint of that it's programmed to export a certain fraction of what it produces. And then the equilibrium is that each cell is optimizing their production, given the production of every other organism in the system.
Except that they're not really optimizing if there's surplus, right?
Well, they could be individually optimizing, but not collectively optimizing. So it might be in the self-interest for one cell to produce a particular metabolite, but because it's programmed to export a very large fraction of it, it doesn't necessarily capture all of its produce. And so maybe this example works. Suppose you have species 1, and it's programmed to export metabolite 1, and it doesn't get metabolite 1 from any other. Nobody else is exporting metabolite 1. It's not endowed in the medium. And nobody else actually needs metabolite 1. It's completely useless to anybody except for species 1. And the species 1 is just programmed to export a certain fraction of it. So it might not be able to, so it's going to lose some of metabolite 1 into the medium just by virtue of being programmed to export a fraction of it.
So just to clarify, so you're saying the surplus is something you observe after you genetically engineer the microorganisms, or in nature without any engineering?
Yeah, so I should be clear. So what I mean is that the export rates are going to be exogenous in this system. We're going to solve this system given exogenous export rates. And then what we're going to do is we're going to do the comparative statics on those export rates to see how that affects growth and population dynamics and things like that. Does that answer?
I think we should go on.
Okay. Maybe you'll, yeah, if it's still an issue, I'd be happy to take it maybe at the
I
end.
think that maybe what Roberto's asking about then is so the equilibrium that the solution to these equations that you're going to find is not an evolutionary equilibrium. Right? If you allowed natural selection to run its course, you would not have organisms exporting unnecessary.
That's right. Yeah, that's true. So what we're looking at, we're not looking. Yeah, that's right. We're not looking at evolutionary equilibrium. We're keeping the amount that we're looking at a short run equilibrium. So these species, the capability of the various species is completely fixed, and we just look at how the system equilibrates via the population dynamics. That's what we're going to do. However, what it does is it highlights also where species might, what directions, what parameters species might evolve to, because you could look at, the population dynamics as a function, you could look at the equilibrium given these particular parameters, and then you could do the comparative statics of changing those parameters and see how that benefits various species. And that's kind of the analysis that we're going to do towards the end.
And by population dynamics, you mean relative abundance of a given production type, right?
Yeah, that's a great point, which I should definitely make clear. So when I'm going to talk about population dynamics, I'm going to talk less about the growth of the consortia. I'll talk about that a little bit, but it's going to be a lot of emphasis is going to be thinking about the relative growth of the different species in the consortia. And so there's actually one of the main results from this is that there's going to be this trade-off, that the species that share a lot are going to cause the consortia as a whole to grow really quickly, but then they're going to be a small fish in a big pond. They're going to have a lower fraction, the relative abundance to the other species in the consortia will be small. So we'll look at sort of both the sort of relative and absolute aspects. Okay, so here's the story. Cells absorb glucose from the medium. They need a particular ratio of metabolites to build another cell. So for example, species 1 might need 100 units of arginine and 346 units of phenylalanine to mitosis, to produce one extra cell. Each cell can metabolize glucose into other necessary metabolites for growth. So for example, 1 unit of glucose might yield 3 units of arginine after being fully metabolized, or alternatively 2 units of phenylalanine. So the key decision, I use teleological language to explain what cells do, because that's how we think in terms of economics. If it offends anybody, it's a slip, so I apologize in advance. So what the cells do, in terms of optimization, is they have to decide where to send these resources, which pathway, metabolic pathway, in order to maximize this ratio for growth, right? Because what they want is 3.46 to 1. Right, that's the optimal ratio for growth. So cells are going to pump out a fraction of these metabolites in the medium, and that's the exogenous part. They're just sort of programmed, they're stuck, they're constrained to pump out this fixed fraction. But they also absorb these metabolites from the medium, and we're going to think we have this sort of diffusion process by how they reabsorb metabolites from the medium. And the fraction that they pump out is hardwired into their DNA, but manipulable by biologists, so that's the comparative statics that I was talking about. Given the inflow and outflow of metabolites, the cell metabolizes glucose into metabolites in such a way that it maximizes its growth. Okay, so here's a cartoon, hopefully that will explain basically what's going on here. So let's say we have two different species, species 1 and species 2. And these are cells in autarky, so that's the econ word for no trade. And what these guys do is they metabolize sugar into green and blue metabolites. And for simplicity, let's say that in order to build a new cell, you need equal amounts of green metabolite to blue metabolite. Okay. Now the difference between species 1 and species 2 is species 1 is very productive at transforming sugar to green metabolites, but it's not very productive at transforming sugar to blue metabolites. Okay, so you could say, for every unit of glucose you get 10 units of that. For every unit of glucose you get 1 unit of that. Species 2 is going to be vice versa. Really good at the blue, really bad at the green. So what does this cell do to optimize? Well, if it needs equal amounts of both, it's going to send its resources down the metabolic pathway of what it's actually bad at. So this is sort of a key thing to note here. It sends its resources to the metabolic pathway where it's relatively unproductive. And now let's go into this system and program these cells now that they're exporting the metabolite that they're relatively good at producing. So, how many people have heard the term comparative advantage? Haven't? Okay. So the people who don't know won't reveal themselves. So that's so I'll assume almost. So I'll explain it very quickly then. So comparative advantage. An agent has comparative advantage in producing X if it has the lowest opportunity cost to produce X. David Ricardo came up with the law of comparative advantage almost 200 years ago. Whenever there's comparative advantage, there are mutual gains from trade. Right? This is like the big result from. This is one of the key results in all of economics, in fact, economists, it's said amongst economists, this is the only thing in economics that's both non-obvious and true. Most of the stuff that we do isn't true, or it's obvious. So the idea of comparative advantage, whenever there's comparative advantage, there's mutual gains from trade. Here he's talking about voluntary exchange. With the bacteria, it's not really voluntary because we force them to trade by programming them to do so. But the point is that there's the potential for mutual gain when there's comparative advantage. So let me go back to this. What I mean by comparative advantage here is if the ratio of the productivity, that productivity I mean how many green do you get for every sugar you use, right? If the ratio of that productivity divided by this productivity is different than the ratio of this productivity divided by that productivity, that means that there's comparative advantage. If the green productivity over the blue productivity is greater than the green over the blue for that guy, then we say that species 1 has a comparative advantage in green, species 2 has comparative advantage in blue. That's true even if even if one species has an absolute advantage in both. Species 1, if this is like for every unit you get 10, for every unit here you get 5, for every unit here you get 2, and for every unit here you get 1. Even though this is better at everything, we would still say that this has a comparative advantage in green, that has comparative advantage in blue. So as long as you have comparative advantage, as long as these productivity ratios are different, there's mutual gains from trade. And you can see this in the cartoon, because what happens is that whereas before, each species is putting all of its resources in what it's pretty bad at producing. Now they're going to put the resources in what they're relatively good at producing. And then they're just going to share the rest so that the other guy gets more resources, and so growth increases. Okay. So by basically, it's just you just have the total efficiency of the system has increased because everybody's doing what they're good at doing. Okay, so trade exploits comparative advantage. Biotic general equilibrium theory predicts the consumption and production of metabolites at a specific point in time. So what we do is we have this model set up. If anybody wants to stay later, I have like the slides that have the math on them. It's basically a linear, it's very similar to general equilibrium theory. It's a linear programming model, and you just solve it using linear programming methods. And what that does is it gives you the consumption and production of metabolites at a specific point in time. Also, since consumption is directly related to growth, basically what consumption is accumulation of bricks. Once you have enough bricks, you have a new cell. We also know the instantaneous growth rate of all the species. So the VGET tells you what the growth rate of all the species are in this system at any specific point in time. So what this is like is it's like forces on a particle, but ultimately we don't really want to care so much about the forces on the particle. We want to know the path of the particle. We want to know the dynamics of this system because it's interesting. This is interesting as well, but ultimately we want to know is how these populations grow. Right? So we want to know the population dynamics. This just gives us the instantaneous growth rate. So we're going to take a first step looking at the dynamics. Then we know all the forces on the particle given sort of initial population ratios, given all the parameters of the model, the productive capabilities, the resource needs, et cetera, the export rates. And we're going to look at as far as dynamics, we're going to look at steady, stable state populations. We're going to define SSS as population ratios of the species. It's the population ratio of the species for which all the species have the same growth rate, and small perturbations in population ratio converge back to the SSS. That's the robust, that's the stable part, right? So basically you have some particular ratio of the species, a vector of the species, of the population of species, and the growth rate of each of those species is going to be the same for all of them. And so that population ratio will just persist. So why do we look at stable steady state? Two important reasons. Most importantly, it's easy. This system could be very complicated. The system could handle an arbitrary number of species, an arbitrary number of metabolites, blah 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 blah. And we want to get some intuition out of this. And if we're looking at more complicated dynamics, it might be sort of hard to figure out what's going on. So we're going to start with something really simple. The states where the population ratio is going to be fixed over time. Right? The second reason is, by definition, it seems plausible that systems will spend much of their time in stable steady state. As long as you're in a universe where there's not that many perturbations, right? What'll happen is typically you'll get these populations that they get into a Bayesian of attraction of a steady state, and they'll just either asymptotically reach that steady state or converge to the steady state, and then they're stuck there. They'll just be there forever, right? So we would think that most systems are going to be in this particular condition. Okay. So BGET is capable of solving in exchange for an arbitrary number of metabolites, inputs and outputs, an arbitrary number of species. So in order to derive intuitions from this framework, we're going to start simple. We're going to explore the model using a simple 2 species and 3 metabolites case, just like the cartoon.
Sorry, I'm going back to your last slide. I mean, maybe we could save this for the discussion, but I don't see where the assumption comes from that, where's the assumption come from that the species have to have the same growth rate? Because you would think that
they don't.
In biological equilibrium, one species just outcompete the other if it has a higher growth rate, right?
Yeah, absolutely. This is
it doesn't seem plausible that systems will spend much of their time in SSS, in real biological systems.
Well, when you consider that the growth rate is metabolite dependent, if the faster growing one is dependent on the slower growing one for a metabolite, its growth rate will drop down when that metabolite concentration drops down. So it's not like the growth rate is a steady number. It's nutrient dependent, isn't it?
Yes, yeah, that's
So,
it.
so the steady state is the state at which they're the same. The fast one that in the same, say in a robust nutrient environment would grow faster. Once it grows enough, it drops that level down to the point where its growth rate matches the other. It's an equilibrium.
And also to address your question, you're absolutely right. We will get situations where we'll have, what do they call it, ecological dominance. We'll have one species that will replace all the others. But the thing is, and in that case, there's no mutualism, basically. But the point of this is to understand the consortia. How do consortia work? So what the model also allows us to do is to say when we're going to have consortia, when we're going to have mutualistic consortia, and when it's not possible. Okay, so we got this simple case. So we're going to say species 1 has a comparative advantage in producing metabolite 1, species 2 has a comparative advantage in producing metabolite 2. Metabolite 3 is glucose, which is used to metabolize metabolite 1 and metabolite 2. We numerically solve this system using linear programming techniques. Okay, here's the results. By the way, I'm very happy to get suggestions and tips on how to market these results, because as an economist, I think the way I think anyway, is we market results often as trade-offs, right? You're trading off one thing to get something else, and that's the fundamental. That's often how we think about things. So we had all these results marketed as trade-offs. You get a little bit of this, you lose a little bit of that. But I don't think that's going to work for our audience. What I've noticed is that biologists and psychologists tend to have really good sound bites that are like an effect, like illusion of control, foot in the door effect, like rock paper scissors dynamics, things like you always get these like really. But so I'm trying to figure out what the good buzzwords are for these. But anyway, it's really important for marketing because I find that the people who are reading this just don't know what we're doing. Okay. So the first, and I've noticed I think 4 words or less. I think they have to be 4 words or less. Okay, so the first one, these are very much work in progress. The first one is comparative advantage drives stability. Okay, so here these are all numerical solutions, and this the x-axis. So ignore panel D right now. Let's look at panel A, B, and C. The x-axis is species 1 contribution rate. That's the fraction of metabolite 1 that it exports into the medium. And this is the vertical species 2's contribution rate. And the colored area is where we have stable steady states. The white area is where no such stable steady state exists. Okay, so let's look at give an example. So for example, if we go like here and we look up here, so this is where species 1, it's exporting, its contribution rate is something like 5%, whereas like species 2's contribution rate is 95%. And so with this numerical, in this numerical example, there is no steady stable state because effectively species 2 is sharing so much and species 1 is sharing so little that species 2 cannot grow fast enough to keep up with species 1. Species 1 will completely dominate that system. Okay? So there's no consortia. It's basically in the limit, you'll just get, you can see the little, a little vial of goop, and you'll end up basically all with species 1. Okay. Now, however, in the shaded area, those are all you get steady states, stable steady states. I'll tell you what the colors mean next. It's not so important right now. Okay, so now what are we doing when we go from A, B through C? We're changing the parameters, and the parameters that we're changing, going back to the cartoon, parameters we're changing the ratio of the ratios. We're effectively changing comparative advantage. So by ratio of ratios, I mean the ratio of the productivity of this to this divided by the ratio of this to this. So the higher that ratio is, we think of the more comparative advantage, the more potential gains from trade. When the ratio is exactly 1, there's no comparative advantage. And when the ratio is less than 1, then the comparative advantage goes in the opposite direction. But we could, without loss of generality, we could just think of 1 and greater. So, here we go. So in this example, that ratio of ratios is 100. As we go from panel A to panel B, that ratio of ratios goes down to 10. And for panel C, the ratio of ratios is 2. And when the ratio of ratio equals exactly 1, it's just white. There are no stable steady states. So essentially, this magnitude, this quantification of comparative advantage is really driving the fraction of the space that induces a stable steady state. Stable steady state is really driven by comparative advantage. Okay, so that's the first intuition that comes out of this setup. The second intuition, these are alternative marketing labels. Big fish, small pond. It's kind of, people know that's catchy, right? Or my co-author he just sent me an email this morning. Good of the group versus dominating the group. And what's going on here, for here, look at panel B. B and D. So for panel B and D, the parameters are exactly the same. This is the exact same numerical setup. What panel A through C all show is the colors represent the log population ratio of species 2 to species 1. So a lighter color means a higher ratio of species 2 to species 1. So let's just sort of look at some comparative statics. Suppose that, let's keep species 1 contribution fixed and see the comparative statics as species 2's contribution begins at 0 and then increases. So as we start here, species 1 is contributing 60% of its resources. Species 2 is contributing, roughly 0, 5% or whatever. And here, species 1 is just sharing too much. It can't keep up. It can't grow fast enough with species 2. Species 2 is kind of free riding on species 1. And so the population crashes. It's all species 2. Now as species 2 contributes more and more, you get around here, you get a stable steady state. You get a situation where both species are able to survive in SSS, but the ratio of species 2 to species 1 is very large. So this is in log base 10. So it's like, like 100 to 1. Right? There, for every cell of species 1, you have 100 species 2s. But then as species 2 contribution goes up, the ratio of 2 to 1 goes down. Right? You get here, the ratio is 1 to 1, and as you go up here, the ratio is below 1 to 1. So the key takeaway here is that the more species, at least within the shaded area, the more species 2 contributes, the lower its total fraction of the population. So it becomes a smaller fish in the pond, so to speak. Smaller relative fish, I have to be very clear, smaller relative fish. Now, panel D is a similar thing going on. So let's do the same kind of process. The colors represent the growth rate of the consortia. And by definition, let me just remind you, by definition, steady stable state is when all the species in the consortia are growing at the same rate. So this color represents the growth rate of all the species. As we go up, from here and upwards, we find that the growth rate begins, quite low, then it gets faster and faster and faster, and then it starts going down again. So what this does is it highlights a trade-off. If species 2 wants to really be the big fish in this pond, relatively speaking, it won't share very much, right? It can be down here, and it'll dominate the population. On the other hand, the growth rate of the consortia as a whole will be very low. If species 2 were to share more, the growth rate of the whole consortia would be much faster, and so this consortia will outcompete other consortia, and as a whole, both species will do better. So this highlights a fundamental trade-off. I don't have an answer to say that one strategy is better than another strategy. It probably depends on its environment. But the key idea is that there is a trade-off. So this is the trade-off that we experimentally test. And so E. coli naturally have pumps that export various metabolites. My co-authors developed a method to program the E. coli to export more or less. And in the experiment, what we have is we have 2 strains. They're auxotrophic strains, which means that auxotrophy is the condition in which a species cannot synthesize the necessary metabolites for growth. Most species are auxotrophs, so humans, for example, we can't synthesize 8 of the 20 essential amino acids. So if you're an auxotroph, you need to acquire some of these metabolites from other sources. So we're using an auxotrophic system. This delta F, these guys need phenylalanine in order to grow. They can't produce it themselves. And the delta R need arginine. The only reason why we're using auxotrophs actually in this experiment, it's not necessary for any of the predictions. The reason why we're using it is because that's the strains that we had available. That's what the technology that they had already built. It's probably actually more interesting if there weren't auxotrophs. But essentially the dynamics of the system are not dependent on this auxotrophy. So we can do we've already done that, and it's basically the same. All the same intuitions come through. Okay, so this delta F guy needs F or phenylalanine, and he exports arginine. The delta R guy exports phenylalanine, right? So these guys need each other to grow. Okay, and here are the results. So ignore this stuff. Let's just focus. Let's focus first on the top of C. So what we're doing here is the experimental manipulation is both of these guys naturally export the metabolites into the soup. The delta R strain is, the amount it's exporting is fixed. And here we're going to vary the amount that delta F exports of arginine. And so what we get is that initially the more it exports, the greater the total growth of the consortium, I guess. And but when it gets too high, the total growth goes down. And this is exactly what we had here. As we increase the contribution rate, it goes up and then down. So we get the same comparative static prediction. The second result is, remember, as we went up, the proportion of the population that goes to species 2 drops, right? The relative proportion. And that's exactly what we get. So this orange guy, we increase his how much he exports of arginine, and his proportion of the population goes down. So we get exactly the trade-off that the model had predicted. How am I on time?
6 more minutes.
Okay. So should I leave it time for questions or just okay? So I'll try and wrap it up. So leave a few minutes maybe at the end. So the last result is auxotroph need friend indeed. So autotrophs will tend to produce less of their export good than what would be growth maximizing due to selfishness. Okay, actually, let me just cut to the chase. This is the way to explain it. When we go back to the cartoon, an auxotroph has a broken metabolic pathway, right? There's some resource it cannot produce that's necessary for growth. So if we were to take the auxotrophic version of this system, we just kill this pathway. But that means that all the sugar is going to go to green, which means it's going to share more green. Same thing for the other guy. So basically, by forcing these guys to be auxotrophs, it forces more trade. When they're not auxotrophs, they have the incentive to do sort of this selfish act. This is a selfish act because species 1 does not export blue. And as a consequence, it's producing metabolites only for itself. If you shut down those pathways, it eliminates selfish options, which could increase trade. we do a numerical example, you could show like, in a simple example, you can get whatever you want, but you could show that auxotrophy makes things worse. But the point is, there are some examples where you could get auxotrophs doing better than autotrophs, which is kind of surprising because basically what you're doing is you're saying that having a cell that has less ability to produce resources will grow faster if it's in consortia and trading.
Specialization in division of labor.
Right, yeah. If it's basically that's a great way. Actually, maybe that's how we should explain it. It's basically forcing specialization. Okay, and so I'll just wrap up. So applications, this could be used to help explain ecological patterns, to understand which species work best, where there's going to be the greatest mutualisms a priori before actually looking in a particular ecosystem. We could predict and explain how microbes might evolve to satisfy these trade niches. There's bioengineering applications, so for example, metabolite production, optimal feedstock utilization. You might want to use consortia of bacteria, might be more effective at producing biofuels or whatever than a species by itself. As I mentioned before, this might also be applicable to multicellular mutualisms. And for future work, what we'd like to do is we'd like to measure the parameters of the model. In particular, we want to get better sense of what the technology of a particular cell is, what its input-output functions are. And there's already work that uses linear programming techniques and using sort of the whole sort of metabolic pathways of a given cell to figure out basically what a cell's productive capabilities are. And also through possibly high throughput cellular phenotyping, basically sequencing the DNA, this could give us a rough proxy of what resources a species needs in order for growth. And then we could also look at some key sort of economic concepts like vertical integration versus outsourcing. Vertical integration would be the idea that perhaps, maybe you're a species and you need to get lots of different resources. Maybe what you do is you differentiate some of your cells. You turn a few cells into a leaf that are really good at getting glucose, some other cells into something else that can synthesize some other metabolite. The alternative strategy would be to outsource this to other species, right? To form a mutualism with a species that's perhaps really good at photosynthesizing or something like that. Blah, blah, blah. And then the last thing I just want to say would be, because I think this might be quite relevant for anthropology. And if you have any, if you know of any papers on this, I'd be very interested, because I haven't heard anything in economics. But I love this idea of the question behind trade versus predation. Do you want to trade with your neighbors or do you want to eat your neighbors? Okay. And so if you eat your neighbors, you get a one-time bonus. But if you trade with them, you get this continuous benefit, right? You can imagine the same thing happens with tribes living next to each other. So I think there's a good Steven Pinker quote, which is like, it's very unlikely that the United States is going to go to war with China because they make all of our stuff and we owe them too much money, right? And so I think it'd be really interesting. I would imagine that I have no idea if it's true, but I suspect that there would be situations in nature where this really is an issue, whether a species would prefer to sort of just exchange resources or just gobble up its neighbor. Okay, so that's the end.
Is there a third possibility, domestication?
I cut out the bullet point. So there's a bullet point that was right here, right? I go up and I'm like, do we want to give out all of our good ideas? And so we were thinking of, and to be honest, I don't quite know what I mean when I say this. We're thinking of like some sort of mutualistic farming, and we're like, oh, can we be the first people to figure this out? And I'm getting all my ideas to the web right now. Apparently there actually was a paper that is the first to find bacteria farming a microbe farming bacteria. Amoebas will take spores and they'll take it with them into novel environments and then farm the bacteria. So yeah, mutualistic, so we were thinking like some sort of farming kind of behavior would be really interesting. We have some ideas about it. We need to talk about to ecologists to see if there are realistic, naturalistic examples.
Thanks very much.